So, I mean, we have a very good day, I would say, long, but with very interesting talks. And if I can make, I would say, a short summary that is a sort of introduction of what I want to ask you, is that in the morning we were talking about adjuvants and maybe more about antibodies. And then in the afternoon we were going towards more the T cells and also the tissue-resident T cells. So in general, I don't know, I have this idea, let's say, that for making a very good vaccine, we definitely need both humoral and cellular immune response. However, I have also this tendency to think that, you know, if we really want a vaccine that is more protecting from the infection, probably we need to have more, let's say, focused antibody response. And for T cells, in a way, it could be better to have T cell to have a vaccine that is more protected toward disease. Now, and I'm always thinking about that a little bit. And my question to you is, if we want to really develop a good vaccine, what do you think we should do? Really have a vaccine that is going to protect from infection, or is more safe to go towards something that is protecting from the disease, particularly after what we have seen from the COVID-19 vaccine, that in one way was heralded as we're going to protect from the infection, but in reality is protecting from the disease. And this has also some, I would say, implication for the social, let's say, well, for the public. I don't know, somebody want to make a comments about that, whether they disagree about that, what you're thinking? You need to help me.
I'll dive in first. So I think your question, or maybe another rephrasement of your question, is how important is sterilizing immunity from a vaccine, and is it a good or bad thing? Because I used to think, like everyone, that our vaccines, we want sterilizing, neutralizing antibody, and you never get infected. I now don't believe, I think that's a myth. I don't think any vaccine actually achieves that. And I would argue, in fact, a vaccine shouldn't achieve that, because nothing will give us better protection than exposure to a virus itself, because it has every element the immune system needs to see to give long, durable protection. So if our vaccine is too effective, we never get to see the virus. And at some point, that vaccine protection is going to wane, and we're left with nothing. So my view now is that a vaccine shouldn't induce sterilizing immunity. It should provide protection completely against severe disease, maybe even mild disease, but allow some degree of infection, enough to boost our immune response so the virus itself acts as the vaccine booster. And what we've seen is if you have that situation, even in COVID, just with a few doses 3 or 4 years ago, none of our people are getting reinfected, and none of them are having vaccine boosters. But what happened was they all got a mild infection. And so maybe the not strongest vaccine at the beginning might end up the best vaccine at the end. So I'm rethinking everything, and I'd be interested to know what everyone else thinks.
Okay. Okay, because I got a mic, I'm going to be the next. Yeah. So I agree with you, basically. You know, so when the pandemic started, so people are predicting that that's keep on changing. So the virus itself is going to be evolving. So it's going to be, it takes few years, but it's going to be like common coronaviruses. And this is basically where we are, right? Still, it's killing some vulnerable people, but still, it's for the majority of people, it's like the common cold-like symptoms. So I think, you know, what we learned during this very big pandemic is that we shouldn't try to achieve neutralizing antibodies itself. So maybe what we have learned is that maybe T cells are really important to protect from a severity. So now I believe that's also my bias too. I think we better induce a very strong and long durable T cell responses. Particularly, you know, people are talking about the CTLs because CTLs can destroy the infected cells. But CD4 T cells are really fundamental helpers for the antibody response plus induce a very durable CT response. So I think, you know, maybe in the future, I think we better induce a very strong and a durable CD4 and CD8 responses to protect from a severity of the disease.
Yeah, I think if you...
You are making vaccine.
making, yeah. Right. Yeah, I think in general, when you design a vaccine, people will targeting the antigens that you can neutralize it and prevent it infection. And then, of course, we also learned that T cell will particularly, all vaccine will induce CD4 cell as well as B cell, right? No doubt about it. Otherwise, you won't be able to make an antibody. But I think if you use the whole antigen, let's say a spike protein, it always have CD8 epitope. You're going to see few of CD8 epitope, right? And that, the beauty of T cell epitope is more conserved. And then you have a chance to cross-protect, and you have heard a lot of this meeting. But if you look at a number of infection after vaccination, I believe that in nature of post-vaccination, you're going to see both sterilization, number of people. You're going to see infected asymptomatics. And you're going to see infected but disease symptomatics, mild. You see the data. And you can see patients that have very severe diseases, and a few of them die, but some disease die more than 50%, right? So I think like H5N1, and then we heard that 50% going to die. So why is that this diverse responses among hosts? It's not only hosts, it's also virus. So the number of inoculation or number of exposure, if you have let's say it's just the theory of war, right? If you have better army and less enemy, you're going to win easily. That's very clear. So if you look at the early COVID vaccine study, you're going to see a significant number of infection protection, right? Because if at that early stage, you maybe have only few number of virus, but the herd is more and more infection, a lot of viral in the community, and people get a lot of virus. So that's why the sterilization failure. So I think making a vaccine, you need neutralizing in the body, for sure. You need a good helper T cell. And if you want aim for durable immune response, let's say only one or no more than two, and you have durable memory, you need to induce a good follicular memory CD4 cell. And at the end, when people working with a too small antigen design, you're going to lose CD8 epitope. So now, So my point is that, let's say if I work with now our vaccine, H5 vaccine, people working on H5 only, but we know that NA is also important. Have a lot of CD4 and particularly CD8 epitope. So I will go with that. I will put more together. So and then in case that you have something diverse, in the virus, you can have some cross protection as well.
Just redirecting a sort of question for you, just sorry. But for example, chikungunya is more, I would say, probably important to protect from the disease here. So what do you think about, let's say, really try to design a vaccine against chikungunya?
Well, I think on the topic of designing vaccines, just wanted to add on to what you all have added. Clearly, the COVID episode actually has rewired our thinking about types of vaccines, like what Nick mentioned. We are not looking at preventing infection, but about how how we can control the progression of the disease. So I think coming to Chick, then yes, I think the old school, it's can we even prevent infection? I think that is actually achievable, but it's just that how sustainable, how soon actually you need the boost. So of course there's the VLP ones, that one seems to be pretty good, where you can even prevent infection. And I think the attenuated one, I think, is a little bit controversial at the moment. But my take about vaccines is there is room for different types out there. And I think the COVID actually showed that there could be certain ones, whether is it the protein, the subunit, mRNA, or even the old school. I think there is room for different ones. And there could be at different times the person will be suitable for that particular vaccine, and then the annual boost will be different. I think I myself, I went for all the different vaccinations. I was, and I still got infected twice. So I think this also shows that it's not going to be totally 100%, but of course each time I was pretty mild. And then coming to chick, maybe I don't need a vaccine for myself because I got infected by chick a couple of years ago. So maybe, I don't know.
I agree everything, but I think I want to think about from different angle, and so the difference with the current vaccine strategy is basically you put a protein just into a person, and when the virus prime it's pick cells, which location, how it's primed. So fundamentally, the cells that primed by the infection and the vaccination is different. That's a problem for the current strategy for like flu, HIV, or at least what I studied. So what's always bothers me is for infection, you have the same experience. We hardly see any good T cell response, except especially CD8 response from the infection for envelope protein. HA and ENF and the spike. For infection, vaccine you can induce both very good CD4, CD8. Why? I think it's probably because the way they prime, and also it's unpublished data because I have a friend who is doing structural biology on envelope protein. So for spike, it's when the virus gets in, it gets just digested so quickly. Actually, it's not much spike actually available for antigen processing, for antigen delivery to cell surface. So let's, from the biology and the structural biology view, it's explanation. So I think that's why sometimes it's important for the vaccinologists to thinking about the virus, how it works, infected. So I think I still believe if we want T cell to help, spike is not a good one. And because if this is the case, thinking about if even you have very good CD8 spike response, but if the virus infected cells don't produce much spike can be presented on the antigen presenting cells, still not going to be killed. So this is something from the lab person for the vaccinologist to think about. And so it's the internal protein, if we want a good one, internal protein, NPM for flu, probably MP and a few others for structure protein for COVID.
I don't know whether there are also some questions from the audience. If yes, please let me know. But following, yeah, there. Maybe while you are coming down, no. Okay, I'm waiting.
Thank you for the nice talks today. I have a very naive question, because today we heard only about peptidic vaccine. What are your thoughts about lipidic vaccine, and do you think could be one of the best way to maybe invest a bit more for better vaccine against any bacterial or parasitic disease?
Somebody wants to speak about lipid? I cannot speak about that. I can tell you immediately.
Well, maybe we can broaden it and say non-protein antigens. Because then we can actually include the carbohydrate structures, which we know on conjugates are very, very effective. But certainly in some of the diseases we've worked in, like typhoid fever, then it's clear that people develop, if they get infected, anti-LPS antibodies, and those anti-LPS antibodies do seem to correlate best, even better than the anti-polysaccharide antibodies with protection. So I'm fully supportive of the idea that we know that happens in natural infection. Why can't we mimic that with a vaccine? And I think it's just people haven't done enough work on, probably you need to conjugate it to a protein to provide CD4 T cell help. But we already do that with our glycoconjugates. So why aren't we making lipid conjugates? I think it's perfectly sensible, but I don't know if anyone's actually doing that right now. But great question.
Okay. No, so continuing a little bit this discussion, while you were, let's say, exposing your different theses, I was thinking we are also not thinking a lot anymore about the potential of immunopathology induced by the vaccine. When you were talking now, for example, about, it could be better to have not spike antigen for COVID and maybe put the structural antigen. But, I mean, we know that there could be the possibility, there have been the possibility in the past of some vaccine that in reality...
Actually published a paper last year in Natural Communication for the spatial omics. So this is the post-mortem sample in lung. And so it's quite clear CD8 is accumulated in disease site. So it must be, they must at a later stage, they contribute the final push. The CD8 is just around a disease area in the lung, not in the healthy, because you have the whole piece. So you can see the disease area and the normal, relatively normal CD8, not much CD4 actually, But they are also report on the transcriptomic analysis, CD4 cells also contribute in the severe cases. So when it's an extreme case, and some people think it's a bystander activation because everything is so inflamed. That's why we're so keen to see the T cell receptor, to see if they are antigen specific or bystander, because everything is so inflamed, so everything, they just gone crazy. They kill each other. Yeah.
Because I'm not sure, were you referring to vaccine-enhanced disease? Was that part of the question?
Yes, I was, because in reality, I mean, in general, I'm in favor of a vaccine that can induce antibody, but a good T-cell response. And a T-cell response that probably can be, let's say, broad, recognizing different viral antigens.
Oh, you mean the vaccine-induced?
Yes.
I think, but you proved that early response.
No, I didn't prove anything. I prove it in the SARS-CoV-2. Hold on. I am.
But I think vaccine, the whole purpose is having it very quick before the virus actually is go mad.
I mean,
control it.
You don't have to convince me that the T cells are playing a good role in the early control of viral infection. But having said that, I think in general, if I'm speaking with people that are asking generic questions about vaccine, I mean, we need also to face the fact that, okay, now for SARS-CoV-2, perhaps we have been even lucky to get good vaccines in general, because the messenger RNA vaccine has been working very well. To be completely honest, the inactive vaccine was also working well. So maybe in that disease, but there have been cases, syncytial virus is the first example, where in reality vaccination has not been exactly going towards the right direction. So in general now, in the future, and shall we think about this? Shall we say, oh no, it was an odd case? Again, I'm not a vaccinologist, okay? So I'm more a T-cell, hepatitis B, therapeutic vaccine. But I'm interested to know what's the
Right, well, I can give you a couple of anecdotes. So when we immunized hamsters, we gave them a whole lot of different variant spike proteins and then challenged them with Delta variant and Beta variant. And we did see that the hamsters that got a gamma spike and then were challenged with the variant viruses actually did do worse than the control animals that weren't vaccinated. So at least for that very specific combination in a hamster, there was a signal, a weak signal, it wasn't sort of dramatic, but that, you know, if you're unlucky, you know, maybe anti-spike antibodies of the wrong sort can actually make the disease slightly worse. Now that's antibody. Now on the T cell side, so I want to balance this out. In influenza, we were looking at mechanisms of protection, and so we were depleting B cells and we were depleting CD4s and depleting CD8s. And what we found is if we depleted the B cells in a mouse and then challenged with influenza, they were protected, but particularly the ones that only were left with CD8s actually ended up with, you know, getting very sick before they recovered. So the CD8s definitely are protective against death or control the virus, but they produce a bit of toxicity on the way. And so I don't know what you call that, because the ultimate outcome is beneficial, but the short-term outcome might be... and how do you test it in humans? And how do you test it in humans? So I think the answer is we need all of the above. And so, and I think we haven't mentioned memory B cells. I think they get neglected. We talk about neutralizing antibody. I think memory B cells are actually far more important, because they can make any form of antibody if you've got them, and they can modify the type of antibody they produce. Neutralizing antibody doesn't change. If it's the wrong neutralizing antibody, it can't fix itself. But memory B cells can actually adapt and mutate and actually make a more appropriate antibody. So I think the best responses we ever see are where we get B cell responses, CD4 and CD8, and I agree, picking just one antigen is potentially dangerous or leaves us vulnerable because for the reasons you mentioned, that the number of epitopes is restricted in a single antigen. So I still like inactivated old-fashioned vaccines like the flu vaccine because they have multiple antigens in them. So we shouldn't discard that, and maybe with recombinant vaccines, you know, we need more than one protein. It's more expensive, it's more difficult.
I believe in normal case like flu B, when there's not much variant, antibody definitely the thing. But for the problem with the flu A, the HA is so diverse, then you definitely need the T cells because it's case by case. If you're lucky, you have a more cross-reactive neutralizing antibody. But the danger is the problem with flu, you have to change. You don't know, you use last year's protein too. So that's why it's important for the flu A to have a T cell on the board. So it's like a secondary in case antibody is completely lost effect. You have at least you still have T cell. I always think antibody for acute infection definitely important. For chronic infection is completely different thing. You need T cells, but acute for COVID but T cell you do need T cell, especially like flu and COVID when the spike and envelope is so variable. You don't know what's come next. Then you do need T cell, at least it's reliable. We know there's always one T cell against the conserved region and can protect you. You only need that one. So I think it's...
Let's say.
The question is also for you. Because I'm interested, you make all the list of the vaccine that you're going to...
Right. No, I think if your question is that has any evidence that vaccination associated in terms of immune-mediated adverse effect. Okay, so that's the question. Let me get back to what I recall is the RSV. If you remember RSV, I think 30 years ago, why RSV 3 or 4 clinical trials have to put on hold and terminated very quickly. And then we learned that lesson when we developed COVID vaccine because of us. Because if you use some antigen that you believe is a protective antigen, but in case it's not, it's induced Th2 responses. And that Th2 response go into the lung, and you have the serious, airway inflammation and pneumonitis. And people, the kid die and then hospitalized, right? So it means, yes, I would say yes, a correct selection of antigen. And then RSV vaccine development has been on hold for 3 decades until fundamental research kicked back in, right? And then we learned that, well, the protective immune response, although it's the same antigen, but you need stabilize, you need stabilize. That's why 2Ps, it come in. So biporin is to stabilize, before as a fusion protein. So I think I would say yes, although your antigen induced immune response, but that immune response was the wrong immune response. Yeah. The second lesson learned that we have is dengue. Right, the first dengue that's approved and has been, I think, implemented in Brazil, and you start to see the kid that, some of them have ADE, So I think it's real. It's real. So the only thing that when you people develop vaccine is that step by step, phase 1, phase 2, phase 3, and even phase 3, you have to have a post-marketing surveillance to make sure that with a larger, a number of events is sometimes it needs like 1 of a million or 1 of 10,000 to see that events. That's why it's a post-marketing surveillance is also critical. In terms of mRNA, if you look at mRNA, I think during pandemics of COVID, we have learned that if you check either ChatGPT or Google right now, so you see that how many people have been vaccinated globally, a number of doses. I would say right now it's maybe more than 13, just a second, 16 billion doses has been vaccinated. And half of them are mRNA. Previously is inactivated vaccine and viral vector. And you see that although we're seeing some of the serious adverse event and death related, right? So that and I think that's real. And one thing that I like to address, and then I keep addressing is that why is mRNA vaccine one of 10,000 or maybe 4 in a million that have young male carditis? Okay, now a lot of hypotheses, none has been proven that is that the case. But my belief is that whatever antigen you use a vaccine, not only mRNA, when you deliver IM is simple, but it's go every places. Antigen go every places. Viral, the viral vector go every places. Recombinant protein, that's not, we don't want to see that actually. What we want is to go regional lymph node, right? And the best way to do is to intradermal or transdermal. I think that's the best way of vaccinologists need to go further. But IM is people familiar, right? And you can do injection very easily. So now our development is almost all the product that in the pipeline going intradermal. Intradermal and needle-free injection, yeah.
Thank you. No, I didn't want to give the impression that I'm, again, a sort of no-vax or like that. I was more thinking about in different pathology, probably in different, in different infection, in different bacteria, we should also really understand the mechanism of the disease and then design, instead, and not be too generic sometimes, nothing more than that. Since you touch, I will say, the way of vaccination, perhaps, again, in the last 4 or 5 minutes, want to touch again on this idea to, let's say, try to induce localized immunity, for example, for respiratory immunity. And the first question somehow is also, I'm interested on CD4 in the, let's say, primary airway, because we have seen some data that where there seems to be in primary airway mainly CD8. So what do you think and what the panel think about, in general, let's say, vaccination that is going to induce localized immunity? If somebody wants to say something.
I think that's a really way to go to fight against respiratory the viruses. The only issue that why we don't go really full swing right now is that we can really use blood samples, like your team really showed. There's a huge discrepancy between local immune system and the systemic immune response. Like that is already shown in even influenza nasal vaccine studies. So in the blood, basically, we don't see much antibody response or T cell response, but still it's protectable. What we're missing is that how do we measure the efficacy of the vaccines after you put like a nasal vaccinations. So I think, that's not really a design of the vaccines, the adjuvant or whatever. I think we need to also develop a method to monitor the immune response locally in the lung or in the nasal cavity to really assess properly the efficacy of the vaccines. I really think that's really key.
So I think the problem still with mucosal immunisation as a primary immunisation is it's still weak at giving a systemic response, which is still ultimately our ultimate protection against severe disease. So my feeling is, and what we're doing is using it as a booster. So in other words, do a parenteral prime to start, and then what we've been doing for TB is we've been doing an inhaled vaccine boost, and we get beautiful mucosal immunity in the respiratory tract from actually doing inhaled. And we're currently doing a human COVID study where we're giving an oral booster, and in fact, that's at least in mice is working, and anecdotally it seems to be maybe doing something in humans. But I'm really interested in how to measure, because part of the problem is if we give, say, an oral booster of COVID, we don't see any change in the blood antibody levels or T cell levels, but in mice we can show protection. So I think that what's lacking, as my colleague said, is good assays to measure mucosal immunity that will convince regulators that our vaccine is doing something.
I think we do start to have assay. Maybe what again is the big issue is what are we measuring and what is the level of protection and how we can correlate that, which is, again, the big problem, for example, for T cells, because there is no question that at the moment, even though, again, I'm totally convinced that the T cells are important for the control, but measuring the level of T cell immunity that correlate with protection is so complicated because there are too many epitopes, where they are. For antibodies, easier. If you want to add few, because the organizer are telling me that it's finished, and I think actually outside there is a good drinking.
With
but
the modern technology, like omics, we can do study find biomarkers in the blood, which I think we might. It's very sensitive. Then if you have a biomarkers or combined signatures, you should be able to, then you can design the kit or something, PCR kit to detect. I think with current, we can't do our traditional measure the T cells, probably in this case. In your case, I think it's really just to spend some money for, probably RNA-seq is enough, proteomic. Then you find the blood markers, then you're done, I think, I don't know. Like what I would do if you have a good cohort, then yeah.
Okay. Thank you to the panel for this brief but intense discussion.